Absolutely. Yeah. Thank you so much for joining this Research Computing Manton Tuesday webinar. We have a number of confirmed and upcoming speakers throughout the month of May. You can check that out on our RC Wiki page. And we also have a symposium, Genomic Innovation in Childhood Diseases, which is starting this Friday and continuing for Fridays at 2 p.m. For the rest of the for the next 4 weeks. We have two keynote speakers. The first this Friday, Munko Lek from Yale, who's going to be talking about his work on limb-girdle muscular dystrophy, and Ann Paduri, May 22nd, who's going to be talking about genetics and precision medicine and epilepsy. And then we'll have panels May 8th from genomic discovery to therapeutics, May 15th on the genomic underpinnings of rare diseases, and also on evolving tools and methods in genomic analyses. Peter, would you like to add anything else to that?
Yeah, I think that kind of sums it up. You know, we're looking into a good participation, and in particular, the opening keynote, Munko Lek, he was going to visit us on site for that symposium. Now it's on Zoom, but we're very excited to have him join this event. And on Friday, David Williams is going to open up, and the following Tuesday, May 8th, following Friday, May 8th, we also going to provide a short update on this genomic initiative that is right now accumulate over 5,000 exomes, and it keeps accelerating right now with the new COVID cohort as well. So that's one update. And in parallel, we also recently secured access to the Trinetics network, and through the network, we have now access to over 20,000 COVID patients from across US with very deep clinical data, and that's already available to anybody at Children's. And we will be using one of the town halls with David Williams to provide a tutorial and information how to access the database. Okay, so I think we're good. We have a crowd of almost 100 people here right now. So Shira, maybe you'll just move forward, introduce the speaker, and let's start our seminar. Thank you everybody for joining.
Thank you. Okay, so today we have, sorry. Okay, so today we have Dr. Erik Melén from Karolinska Institute in Sweden. Erik is a professor of pediatrics and a pediatric allergist at the Karolinska Institute, as well as at the Sachs Children's Hospital. And he's going to be talking today about he's going to be talking today about the length of pregnancy altering child's DNA.
All right. Hello, everyone. Thank you indeed for the kind invitation, Shira and Piotr and the others at the Children's Hospital. Really pleasure to be here, although I'm in Stockholm and you're in Boston. But these webinars are really great experience also here at KI. We use it a lot now, and it's a good opportunity to share data and new papers, etc. So again, very happy to be able to present some of our work here. As Shira introduced, I'm a pediatric allergist. So I've been to clinics today. I do see children with severe asthma, severe allergies, but also with pulmonary problems. And research-wise, I'm a PI of a pretty large cohort here called BAMSE. I'm a genetic epidemiologist and try to understand really the origins of asthma, lung problems, allergies, etc., and the consequences long term. Today I'll be sharing some results from a recent paper, and I'll talk about length of pregnancy altering the child's DNA and results of new epigenetics studies. So before just getting into the research, I saw Benji signed up. Hi Benji, good to see you. I was this picture taking a while back now. I did my postdoc at the Channing 2009. It was really a fantastic experience. I was accompanied by wife and my two kids, twin boys, Victor and Jacob. They were at the time 5 months when we arrived, maybe here after the summer, 8, 9 or something. So that was a fantastic experience, really. I was really looking forward to catching up again and visiting Boston maybe this year later, but that has to happen some other year, I guess. Anyhow, length of pregnancy alters the child's DNA. This was the press release that came out a couple of weeks now when we had the paper. What we did was really study the pregnancy, length of pregnancy, and methylation level, and how that tracked with gestational age, and then followed during childhood and up to adolescence. And the overall topic, obviously I'll get back to the paper, is can epigenetics inform us about development? This slide just shows the definitions of preterm birth on extremely preterm, very preterm, preterm, etc. It's a bit old now, and maybe the borders are constantly moving to the left in this picture. But that's really central in this project, and also for the understanding about development. And again, speaking to a pediatric audience here, I'm sure you're all very aware of this situation. And when I think about epigenetics, again, linking the genes and exposure, most studies have been conducted on the methylation. So on the change of a methyl or addition of a methyl group to the DNA C, where C lies adjacent to G. Also, obviously, histone modification, but methylation is the most studied factor so far. And again, even though this is basic, apologies for the more experienced people in the audience, but again, when we address these questions, why is this important? Well, in the CpG island, in the promoter sequence, if it's not methylated, the gene can be transcribed, fully active, whereas in case of full methylation, the gene can be completely shut down and suppressed. So it's a very powerful mechanism by which the cells can direct or orchestrate gene activity and protein production. And interestingly, again, linking the exposure with epigenetics is, today we know that factors like dietary factors, folic acid intake, do alter the methylation levels. Traits like obesity also seem to change methylation. Smoking is one of the most well-studied examples here, and I'll just show one study that profoundly found strong influences. Aging is perhaps, together with smoking, those are the two factors really driving the epigenetic signature and DNA methylation status. Also, other exposures like pollutants. And again, the theory is that if you change the DNA methylation level, you do alter the gene activity and gene expression. And this old famous paper published in 2003 is also again showing the power of this epigenetic signature. In this case here with all transgenic mice, they all genetically exactly the same, whereas the mothers were fed with different folic acid intake, and that in turn, the mother's intake changed the methylation level in the key agouti gene that in turn determines the color of the fur. So it has consequences, obviously, also physiological readouts, these methylation changes. I mentioned smoking. This paper came out 3 years ago, I think now, DNA methylation in newborns from the PACE consortium, where we looked at maternal smoking during pregnancy and particularly sustained smoking, and studied the DNA methylation changes across the chromosomes using the available 450K Illumina chip. And here's the log 10 minus p-value, so these are highly significant. Hundreds of bonferronic significant changes following maternal smoking compared to non-smoking during pregnancy, and also like the dogma suggests, these sites also influence gene expression levels. we were able to find persistent effects up to school age. So meaning that if the mother was smoking, that induced a DNA methylation change in the newborns, and that level stayed more or less unchanged up to school age. So these are pretty substantial findings, I would say, and genes are involved in asthma, CBD, cancer, and neuropsychiatric disease, birth defects, etc. They're also well-known detoxifying, involved in the in cells, and enzymatic activity to detoxify the compounds of tobacco smoking. And if we put it now in a picture where we understand that maternal smoking during pregnancy is obviously affecting the fetus in utero, and that induces a widespread DNA methylation change, numerous sites across the genome. Some of those changes do influence gene expression. It could be more genes, gene activity, or lower, depending on. Key question is obviously, is that going to lead to disease such as asthma or other diseases? I mentioned the other exposures also can affect DNA methylation. Here's just one example. We've been extensively looking at air pollution levels. We relate that to asthma development, lung function development, etc. And that's a pretty clear association also here in Stockholm with relatively clean air. With the higher exposure, the lower the lung function development up to adolescence. And here we were also able to find associations not at all as strong as for the smoking, but we do find significant associations between exposure and methylation changes. And we do a lot of pulmonary research here in my group. So and some of these genes are well known, Notch1 for instance, not well known involved in different lung diseases. So there's certainly evidence that the exposures do change methylation levels. So what about disease? I'll take one example here, asthma, also as a background, again, setting the scene for the epigenetics and the complex traits. Is asthma actually associated with changed methylation levels? In this study, we worked with cohorts, European cohorts, more than 5,000 subjects, 450 again, with really try to do replication efforts and run functional analysis to understand the meaning of these changes. And this paper came out 2 years ago now. It's led by Gerard Koppelman and myself, and it was through the MEDAL study, European collaboration on asthma and allergy. And we started by looking at whole blood and simply said, all right, are there any differences in methylation levels between asthma cases and asthma controls or non-asthmatics? And here we find I think 14 CpGs significantly, and they all replicated very nicely. But when we look at the absolute level, it's actually quite small absolute effects. So a couple of percent difference. Here's the beta value for the methylation level. This is the whole blood, asthmatics, and controls. And you can see that there's a couple of percent difference. But then we teamed up with the Catherine Laprise and her colleagues from Canada, and they had isolated eosinophils. And we looked at the same CpG sites here, as you can see, and in isolated eosinophils, that small differences in peripheral blood, in the mixed blood, was substantial in the eosinophils. So that really signals that some cells, some immune cells, or it could be other cells certainly, really there are profound differences between cases and controls when we look for epigenetic differences. And I think that was very, very reassuring to see. Eosinophils also, as you know, are highly linked to that of the asthma. Again, a key feature here is to go associate methylation with expression. And we used the BIOS consortium to do that, and in a large study on more than 2,400. And I think also quite interestingly, the epigenetic pattern that we found that being associated with asthma that pattern was also very strongly linked to gene expression signatures known to be involved in eosinophilic activation. So that's one thing. And the other is that it was also linked to naive CD4, CD8 T cells, meaning that the virally induced immune responses. And I think that part of the study was also key to understand the importance of these epigenetic changes. So if we conclude that, again, as a little background, we found CPGs consistently associated with asthma across cohorts, strongly driven by methylation levels or lower levels in the eosinophils, and that if you could use that in the clinic, I mean, that's a major promise to go for a clinical test, for instance. But we also know that isolating eosinophils for a diagnostic is not a trivial thing. We see here evidence that an early life shift from naive T cell population for effector memory CD cells suggests also, host virus interaction. Certainly, we all know that for asthma, rhinovirus, RSV, and other viruses are really triggering the asthma. So that is a little background also for why I think it's important to still identify, explore these epigenetic signatures related to complex diseases or traits that we know of. And I mentioned age, and I'm sure many of you have seen Harvard's age calculator, where you can take a blood sample from someone and tell his or her biological age. And if you have bad luck, your DNA tells you that you're actually older than your chronological age. That's not what you want. You want to stay young, not forever perhaps, but still be young biologically. And that has been used also in many of the studies. Here's a paper in eLife from Karolinska colleagues where they looked at the health risk and mortality, and really found that the aging, if you have accelerated age, that is associated with also increased risk of mortality in adults. So certainly, and I know there's been many studies like this. Colleagues across the street from you, from the Channing, published this paper last year on epigenetic age acceleration associated with allergy and asthma, and perhaps many of you were involved here also. But again, the DNA level and calculating the epigenetic age and the acceleration, and that was actually then associated with the likelihood of being sensitized and also for asthma. Although the odds ratios are not very strong here, but that's clearly also signals that the epigenetic signatures is associated with the clinical relevant outcomes. And again, if we could come back to the epigenetics, if that can inform us about development, and let's now focus also on the preterm birth and the low gestational age. Other colleagues from this PACE collaboration published, I think it was last year, also on epigenetics on the association with birth weight, and very strong associations were found also in that study. So birth weight and intrauterine growth also seem to track with epigenetic signatures. So there has been certainly evidence before our study. Also, I'm choosing this study because again, looking at the pulmonary phenotypes here, alteration in gene expression and DNA methylation during marine and human lung alveolar septation, and again highlighted that methylation is an important regulator of gene expression, and here they also found that is associated with a normal and abnormal septation and lung development. Obviously, for preemies, the bronchopulmonary dysplasia is a key factor here, and whether this is an important mechanism for long-term health outcomes perhaps remains to be discovered, but still important findings. And we know from this is a study from the Karolinska showing the young adults with a history of BPD truly have lower lung function in their young adulthood, and they resemble the CPD phenotype to that extent. And again, the role of epigenetics here for the early life changes and long-term health consequences obviously needs to be discovered. There's some evidence suggesting that these might be a very important factor. All right, let's now get to the actually paper, epigenome-wide meta-analysis of blood DNA methylation in newborns and children identifies numerous loci related to gestation age. It was one of my PhD students, Simon Kibede-Merid, who wrote the paper, and it was published in Genome Medicine a couple of weeks now. It's a PACE consortium paper, and I'm grateful for the collaboration in PACE. Again, many of you might be also involved. I see Project Viva here. But we collected 26 cohorts for this collaboration from 13 countries, 11,000 samples, and they all had Illumina 450K. Nowadays, you'd hope that there'd be 850, the EPIC array, or even sequencing, but at the time we had 450. But certainly the whole of Europe and the US participated, also Australia down there. And again, Stephanie London leads this PACE effort. Also, Janine Felix and others from Rotterdam is involved. And it's really been a fantastic endeavor and effort to bring together cohort scientists working on prenatal questions, research matters, and the epigenetics around that. So this is from the NIEHS webpage, and that Stephanie is hosting. And have a look. And also, if you're interested in further collaboration on a wide range of exposures, diseases, outcomes, so let me know or send an email to Stephanie. All right, in this paper, we had the aim to investigate the association between gestation age and DNA methylation in newborns and older children. We had, this is our cohort, sorry for spelling that out here in particular, but Bamse was one of the cohorts and many of these cohorts. We had, again, DNA methylation, and we linked that to gestation age in a number of days at birth. So here, typically, you'd use a robust linear regression model and followed by meta-analysis. Here we want to really be sure that none of the potential confounders or potential other factors that could influence both preterm birth and gestation age, obviously, and the DNA methylation, that we didn't include those factors, or that we didn't include children whose mother suffered from preeclampsia, C-section, induction, and diabetes, hypertension, etc. So we wanted to be clear that this is only studying, we're trying to really focus on the gestation age rather than all the complicating factors around that. And certainly we also adjusted for maternal smoking, for instance, sex, and a couple of other factors that are known to also potentially influence DNA methylation. And what did we found? Here's again the Manhattan plot, the -10 log p-value across the many chromosomes, and each dot is one CpG site as usual. And you can see almost the significance level of the maternal smoking paper. So here 10 to the -100. So there were really substantial findings. We found 8,899 CpG sites mapping to almost 5,000 genes. Most of them had actually, if you look at the volcano plot, had a negative effect or a negative estimate, meaning that the longer the gestation age, the lower the DNA methylation in that particular CpG. If we said, well, we want to really focus on genes which have at least 3 CpG sites, we found 1,200 CpGs and mapping to 300 plus genes being associated, and again slightly more negative effects when we look at the direction of effect here. But as you can see here, similar to maternal smoking, is a very widespread effect across chromosomes, across genes. Okay, and then the first question, okay, is this something that just happened in cord blood in newborns, or can we detect that earlier if we would use other biological specimens? Or can we also detect these differences in other tissues? So here we worked, we took our top hits, those CPGs that were really consistently on the 3 CPG site selection, here 1200 76 CPGs, and asked whether we also see differential DNA methylation in fetal lung data. And here again, I'll like to thank Boston colleagues Alvin Coe. I don't know if you're on the TC today, but Alvin helped us with the fetal lung analysis. Thank you again. And we found out that among the 1200 CPG sites, 151 were significant also during early, and we're talking about earlier human lung development here from aborted fetuses. There's certainly an overlap between the cord blood data and the lung data, reflecting cross-tissue and earlier effects. The other thing, we also had access to fetal brain data that was available online, and here we actually saw even larger overlap between the significant cord blood and fetal brain. So for this, it seems to be a general finding that some of these CPG sites do track, or the level of methylation do track with gestation age across tissues, not just cord blood, but also fetal lung and brain. And here, the next question is whether these CPG levels that children are born with, are they stable across the childhood and up to adolescence? And this actually, I think this is one of my favorite slides of the whole project. We actually found, again, out of the 1200 something key CPG sites here, we found that 83%, majority did change with age from birth up to school age, whereas 17 were stable. Now that means that, again, you'd expect that a good proportion of these CPGs are changed just because the fetal cells in the blood would die away or vanish from the circulation, and they would be replaced by mature immune cells in the infant, in children, and you'd expect this pattern that there would be a very dynamic change here. But if we start with looking at the 70% of our identified really significant CPG sites that associated with gestation age and preterm birth, they're stable across childhood. Now this study is a Spanish study called INMA. We had birth data and early childhood at the age of 4 or 5 years, and you see for this CPG site it's very stable. This is ALSPAC. We had 3 time points: birth, childhood at 6, 7 age, and adolescent at 15 or 16. And you can see here it's completely stable CPG site level, meaning again that level you're born with, depending on your gestation age, that will track with your age at least up to adolescence. Whereas other sites again had a very dramatic change. So if you were born with a rather high level here, that level dramatically changed and lowered by the age of school age or something. If you look here at the ALSPAC study, most of the levels seem stabilized by school age. So you see rapid, major change from birth to childhood, but very little different between school age and adolescence. And some of them again were decreasing. These two were just good examples of how that might look. And again, others were increasing, so starting low and decreasing with time. And if we do gene ontology and look for the biological process and pathways here among the significant CPGs, perhaps not too surprising, lots of cellular processes, regulation, development, organism structure development, regulation, signaling, etc. So morphogenesis. And we know from previous studies that epigenetics is a very strong mechanism, or a very important mechanism regulating development in utero. So that's kind of expected, but it's also good to see that that's actually what we see. If we do look at some of the key terms here, cancer shows up. Cancer is about development and growth, so it's not very surprising. But also some of the immune signaling pathways here with upon CMV infection or other cancers, papillomavirus, etc. So really suggesting a strong link to immune responses and obviously development and growth. So if we conclude here this paper on the gestation age and methylation, we found actually a strong association between gestation age, and we can also change that for preterm birth, and we find also very strong data. And methylation status in newborns. Maybe I didn't show that slide actually, but it was not just limited to preterm and comparing preterm with term. Also within the term babies, if you compare week 37 with 38, 39, 40, 41, we see very significant changes also within the term group, which suggests really that these epigenetic changes are not just about very preterm events, but also these are taking place also, within the range of term period again. So this is a very consistent finding. We see enrichment for processes critical to development and functionally related also to human disease. Certainly, many sites capture epigenomic plasticity of development across tissue. I highlighted the cord blood, fetal lung, and fetal brain. With the longitudinal analysis, again, I think that's a very important component of this paper. We showed that the gestation age associated CpG had a very rapid change, mostly during early childhood, and then stabilized by school age. But the subset, some 17%, were stable, that did not change over time. And the level with which you are born, you'll have to live with that, so to speak, at least until adolescence. So that was my conclusions and my presentation today. Here's my group. It's a big group as of today, but we have people, postdocs, researchers, PhD students, and bioinformaticians who's certainly helping out. And with that, I'd like to thank funders, etc. But happy to take questions and initiate discussions.
Thank you so much. Fascinating. We have a question from David Williams. I'm going to unmute him. And please, anyone else, raise your hand and we'll unmute you so you can ask questions yourself or type them. David, are you able to speak?
Yes, I think I can. Can you hear me?
Yes.
Yes.
I was curious. I did my doctoral dissertation in the association between any number of factors and adverse birth outcomes. I was surprised in that to learn, or at least that a lot of people believe that the period of pregnancy during which a woman smokes has a strong role in determining the kind of outcomes, the kind of effects. And was that true for genetic effect as well?
It's a very good question. We try to address that in that pace, and even I think we had 6,000 subjects, children. The most profound effects were seen for persistent smoking throughout pregnancy. Off the top of my head, I can't remember exactly, honestly. I know that there are the effect of smoking are very strong. So there were some effects also. If for example, I mean, the classical situation is that someone or a woman smokes finds out that she's pregnant and stops smokes. And the question is, is that also, can we see signatures in newborns? Not at all as strong. Not at all as strong. I would have to look up the details. Sorry for that, but it's very clear that the strong effects was clearly on the persistent smokers, but certainly some effects were seen also. I don't know if anyone else knows by heart the answer to that question, whether very early smoking, early quitting might have persistent effects on the newborn DNA methylation. I'll look it up for you.
All right. Well, thank you. Thank you very much.
All right.
We have another question from Heidelise Als. I'm going to unmute. Are you able to ask your question, Heidelise?
Hello, thank you for your good talk. I don't see myself at the moment, but that's okay. Is extrauterine experience in the NICU involved in modifying the methylation patterns you see?
Can we repeat the question, please?
I also wrote it down. So is extrauterine experience what the baby in the NICU, in the newborn intensive care unit experiences, is that involved in the methylation changes you may see post-delivery into early childhood?
That's a very good question right there. I mean, it's a very special environment for the very preterm baby to be in a NICU compared to being in utero or a moderately preterm baby. Honestly, I don't know. I haven't seen any study that would link the NICU environment, specific NICU environment with methylation outcomes later. I wouldn't be surprised that some of the more severe infections with the timing of the nutritional aspect or introduction of when we introduce formula or other nutritions, antibiotics or treatment could potentially certainly come into play here. But I haven't seen any studies. I don't know the exact mechanism here. In our study, we could say that some of the changes were profound. Certainly, it looked on average that there were very strong effects, and we think that that's mainly due to cellular differences, so that the newborn cells, then the infant cells, really are removed from the circulation and then are replaced by immune cells from the child. But the stable CPTs, they were stable across childhood up to adolescence, meaning that they were obviously not that much influenced by environmental factors or external stimuli or cellular compartments or different cellular phenotypes in the children. Did that answer your question?
Yes, I was more interested in the sensory experience of the preterm infant. And in follow-up to this first question, at your own hospital goes on actively what we call ultra-early intervention, neurodevelopmental, MIDCAP for short intervention, that seems to have good outcome effects, including brain changes that our group has studied. We have not had a chance to look at the methylation patterns, always wanted to, but couldn't get there. So, his name, Rossi, someone in Italy, has begun to look at that. I may not have his name correctly, but I would be interested in your speculations.
I agree. I think that would be very interesting. And again, I think in general we find very so more or less any environmental factor can influence, or any trait even. I mentioned BMI, aging obviously, but BMI certainly. There's also some evidence that if we look at newborn, in another study, also case study on asthma, we looked at newborns and try to relate that to later childhood experience. And if we combine the two asthma studies, some CPT sites seem to be there already newborns and can affect later risk. Others seem to be acquired changes. So in our study, in the Lancet paper, we could not find those changes already at birth. So there must be something in the environment, or the viral infection, or something else. Also, we know that SNPs genetics, maybe Benji, better here about genes, SNPs regulating DNA methylation levels, MEQTLs. That's absolutely also happening. So it can be both ways.
Thank you.
We had a question from Benji.
Yeah, so first of all, really nice talk. Congratulations on the paper. Also, it's excellent work. I can start my video too. It's
nice.
Hi, good to see you again. So first, just to comment on what you were just saying, and to ask follow-up. So we and others have methylation QTL data, and I'm wondering if you've had the opportunity to look at whether or not the loci that show these gestational age effects are less likely to be under those methylation QTLs or the like. And if not, if you haven't done that yet, it would be, I think, interesting to look at. And it kind of follows into the question I was going to ask. Your favorite slide, if you would say the one where you show that 83% of the variants of the methylation marks change with age. So are you suggesting that if you're born prematurely or anywhere along the gestational age spectrum?
Yeah. It's a spectrum.
And right, and that drives your methylation in one direction over another, that with time that regresses to the mean, that sites that were heavily influenced by gestational age eventually are going to regress, or are these permanent? And you were talking about other marks.
Yeah, I'm going to pick that. Since this is my favorite slide, I'm going to show it again, if I may. No, I think we're seeing two different patterns here. One is regression towards the mean. If you look at the left-hand side here, where my arrow is, that's clearly you start off high, and then the levels will normalize by childhood, and then it's a stable level by the time you reach adolescence. Again, this is not all the CPTI. This is only those who are very robustly associated with gestation age at birth. So here you'd see the regression towards the mean, or if you start high and end up lower, or if you start low and by school age you reached your balanced, or this is some form of equilibrium here. This is where you end up. But for these 70%, there's no change. So the level you're born with, that those are stable across adolescence. And so in this analysis, there's same subjects being measured at birth and childhood and adolescence. So it's not different. So in that setting, for those, they're stable.
So this ends up being about 300 of the marks that you identified. There's just a permanent mark of gestational age, you think? Or yes. A relatively permanent mark, at least until adolescence.
Yes, that's what we see. And if we do run pathway analysis, they similarly associate with some of the diseases I highlighted, or infection diseases, autoimmunity, etc. Now the key question is, and no, I guess no one has proven it yet, is that's just suggestions that might be associated or somehow involved, but I'm not saying anything about causality here. because I don't know. And we don't know whether these, signatures here we are seeing are actually causally, related to any of the, long-term health effects of being born prematurely, for instance.
Very good. Great. I just, so I'm going to have to get off a few minutes early, but this was a great talk, and I'm glad that I could see it. And I'm sorry we couldn't see each other in person.
Next year.
Absolutely.
We have another question from Alice Lee. Alice, you should be unmuted. Alice.
Can you hear me?
Yep. Yep.
Yeah.
Yes.
Oh, hi, I'm Alice Lee from Children's. I think it shows eosinophils specifically shows some DNA methylation change in some of your slides. Am I correct?
Yes,
so
correct.
Comment why those cell types specifically show that pattern, or are there any other cell types in blood show similar level of difference in terms of DNA methylation?
Absolutely. If I may, I'm gonna also share this again because it's a very informative slide. It's a really good point. I think it's also a really key aspect of epigenetics research. So looking at blood or looking at any tissue, we must know that this could be cell specific, tissue specific. And we know in other works that if we look at all the different immune cells in the peripheral blood, monocytes, the T cell, B cell compartments, neutrophils, eosinophils, etc. For some CPGs are very stable, others are highly varying across the cell types. Now, in this study in the eosinophils, we were particularly interesting to find or to see that the eosinophils has such a major differences, also because we know that for asthma and in kids, they typically have allergic asthma. Eosinophils are the culprits. So we know that there's a strong engagement or eosinophil activity be linked to allergic asthma. So the question here, we partly what we see in the peripheral blood is can be explained by the eosinophils, the difference in eosinophil. Now the key question here, which I don't have the answer to, is whether this finding here is also leads to activated eosinophils. And because one thing is if you have eosinophils in peripheral blood or in the tissue, that's one thing. That's what we typically use when we look at cell counts and we classify eosinophil asthma, for instance, peripheral asthma, peripheral eosinophil count above a certain level, 0.5 or 0.3, or in the sputum eosinophils. But what do they do there in the sputum or in the lungs or in the peripheral blood? Are they activated? This is quite interesting, and if one could, if that's the case, that the methylation signals is also part of the activating these eosinophils, making also releasing granule, etc. That potentially could be a target, certainly, because we all the steroids and all the anti-IL-5 treatment, the biologics for asthma, is about targeting eosinophils. Not all, but a lot.
Okay, but you haven't, because of the no knowledge, you haven't checked actually other blood cell types.
We did not do that in this study, no. But what we did was to check. We know, in other studies have looked in general, neutrophils, eosinophils, monocytes, etc. And there we checked roughly, and so some of the sites seem to be different across the cells. Others are very stable. But the key thing here is we see the difference in asthmatics. That's the key. In general, we wouldn't expect these CPGs to be different in maybe eosinophils from asthmatics and non-asthmatics. But the key thing here is that we get the eosinophils from asthmatics. And unfortunately, we do not have access to purified neutrophils or monocytes or anything else from asthmatics.
Okay, thank you.
Thank you. Are there any other questions? I don't see anyone with hands up. If there are any other questions, please let me know. Oh, I see another one from Heidi-Louise. I will unmute you.
Or if you have questions about COVID-19 approach in Sweden, you're welcome to, but maybe we should take this first.
Thank you. My question is, it may be too naive for you. Do you see that 17% of stable methylation 2 reported across the range of prematurity, or are they more predominant in the earliest or the later ages of prematurity? And do you see them at all ever in full terms?
It's a very important, good question. In general, there were very clear overlap between findings, the early preterms and in the moderate or late preterms. So these results did not seem to be driven by the early preterms only, or the very preterm. So there's a good overlap. Honestly, we did not check the 17% of the very stable ones if they were more driven or represented by the very preterm babies. But general finding was very clearly that these findings operate across gestation, and we looked at the term babies only. Very strong signals also, by day, length of gestation during the term period.
So the terms have the same 17% of methylation findings, or
That's a very good.
Or is it unique to preterms? Is it specific to preterms?
Honestly, we did not look specifically for those 17%, whether they were unique to the very preterm only. But we looked in general, and there were very strong overlap. So none of the results, what we saw, were driven by the very early preterms.
Right.
So very
but you
strong.
also saw them in full terms?
What we compare here is methylation level with the length of gestation. So we meaning relate the level with the length of gestation. So we also did preterm versus term, and we found lots of differences as well. So yes, we found these differences even in the term babies. So if you look at the range week 37 to week 42, we looked at that by day difference. There were very strong signal. Yes.
Okay. Thank you.
Thanks.
All right. Any other questions? Okay. Well, thank you so much, Erik, and thank you everyone for participating.
Thank you, Shira. Thanks for listening, everyone. Have a good day and stay healthy.
You too.